Good morning, everyone. This video is going to walk you through how to complete your unknown number 1 lab report. It is posted under Lab Unit 1 in the unknown number 1 folder. So essentially what's going to happen is I am going to email you with the number of your unknown, which you will record on this sheet. And then I will send you at least 2 pictures. A picture of a colony, as if you had done the streak plate technique in lab, what kind of colonies you would have gotten. And then I will send you a picture of the gram stain for that unknown organism. Because remember, we're doing this virtually. So normally if we were in person, you would be doing the streak plate, you would be doing the gram stain, and you would be getting this data yourself. But since we're virtual, I am essentially giving you your data, okay? So the 8 possible species that you might have are listed right here on the document, okay? And you're trying to figure out which one you have. So you are going to look at the picture of the colony that I send you, and you are going to fill out table number 1 using the using the information we've learned previously. Okay, now make sure you are always using microbiology terms. Okay, there were terms associated with colony morphology that I expect to see here. And you're going to include a picture of your colony as well. That way I can see it, I can make sure you're using appropriate terms, okay? And then what you do is you switch over to looking at your gram stain slide. So again, notice I'm telling you exactly what you're looking at. All right, you're going to tell me the color. What does that mean? Okay, because you're going to learn that when we do staining, you know, one color means one thing, a different color means a different thing. So you're going to tell me if your organism is gram positive or gram negative, and then you're going to use the terminology that we've talked about to describe morphology and arrangement of cells. Okay, so for this whole thing, you're going to need to reference back to the lab notes and the lab PowerPoints and our discussions over these labs to make sure you're using the correct terms. Okay, remember, colonies are on agar plates. Cells are what you see under a microscope, because a colony is just a mass of millions and millions of cells stacked on top of each other. So there's different terminology for cells and different terminology, excuse me, for colonies. Again, you'll make sure to include photo so that I can ensure that you are, you know, interpreting your data correctly. So then what you're going to do is go through the gram stain PowerPoints and look at the pictures of each organism, and you are going to tell me if each organism is gram negative or positive, and then you are going to give me their morphology and arrangement. The reason for this, okay, is because you are comparing your unknown organism to the 8 that you might have. And so if you look at this who can it be question, notice you're looking at the gram stain data, okay, to be like, all right, well, my organism is gram negative. Well, so are, you know, these organisms. So I must have one of them. And it's just a process of elimination, okay? But you're also going to want to do that by comparing it to your colony morphology data as well, right? If your colony is clearly convex and Bacillus subtilis is not, then you can rule out as your unknown organism, okay? So that's why we're doing that colony morphology table in class. It's helping you figure out what your unknown organism is going to be. And all you need to answer that question is the picture of your colony, the picture of your gram stain, and then the other information I'm having you fill out in these tables, okay? This is all available on the PowerPoints on Blackboard. So then you're going to get down to differential stains, okay, which we'll be talking about in lab soon. and you're going to learn that only certain organisms will have the acid-fast stain done on them. Only certain organisms will have the endospore stain done on them. And so you're just going to go through here and tell me yes or no. But notice, y'all, I am being very picky. You put a Y or an N in each of these boxes. Don't spell out yes, don't spell out no. If I tell you what I want in the table, make sure that's what you give me. This is how it's going to be in nursing school and other programs. Sometimes you have to just be very specific. So you will lose points if you don't follow directions. Okay, it even tells you right now, or right here, every box should have a Y or N for yes or no. So then you're going to say, okay, do I need either of these stains done for my unknown? Okay, now I'm just going to make up information here. Okay, this is false information. We're going to pretend. If you were told me that E. coli gets the acid-fast stain and you think your unknown organism is E. coli, then you would come down here and you would write conduct. Okay? But if you think you have Pseudomonas and it doesn't get the acid-fast stain, you would put Na. Again, notice I'm telling you what your options are for this box, and you have to fill out every box on this report. All right? Now, if your organism does not need either of these stains, that's perfectly fine. You don't have to put anything here, okay? You can put Na, doesn't matter. But if you do need one or both of these stains done, you write here which one, you send me an email, and you say, Chelsea, I have unknown number 3, and I need the acid-fast stain. And I will then send you a picture of the results, okay? You'll put it in this box, and then you will tell me what your organism is. You will write it out completely and properly according to the binomial nomenclature rules that we've discussed, okay? So I will give you the colony and the gram stain. You will request a picture of the acid-fast stain or the endospore stain only if your unknown organism needs it, okay? So if you come up here and you tell me that Staphylococcus aureus doesn't need either of these stains, and then you tell me that your organism is Staphylococcus aureus and you've requested the endospore stain, you're going to lose points because all of it has to line up and make sense. If Staph aureus does not need either stain, then don't request a picture of that stain, okay? So you're basically just taking a lot of this stuff from Lab Unit 1 and putting it together here to identify an unknown organism. And I'll be sending you your pictures within the next day or so. All right, y'all, read over this, watch this video, and come to class with questions. Have a great day.